See, you have 15 hours of revision to do. Test after test, your revision pace will increase. Now you might have felt that you are not able to complete the entire revision, even though the notes is yet not complete. So with every test, the speed with which you will revise will increase. How do you go about revising? Start with syllabus map, physics, chemistry, biology, all three. Go to level 1 notes, physics, chemistry, biology. Go to one perspective of all three, glossary, physics, chemistry, biology. Then the next perspective, physics, chemistry, biology. Then the next one, all three subjects. Next one, all three subjects, like that. Not syllabus map, level 1, perspective notes, everything of one subject, then move to the next subject. No. You understand? Go into the revision like a spring. You complete one full circle of one level, that is syllabus map, then go into the next circle, level 1 notes, all three, next one, next one, next one, goes like that. All right? In 15 hours, today probably, you did not have 15 hours totally. You had around 10, 12 hours maybe. Yesterday you did not get full 8.5 hours. You got 6.5 hours, no? 6 hours. So totally you had about 12 hours of revision time. In 15 hours, let's say you do some part of revision. You have done some part of revision today. Next time, the same material that you have revised for today's test, you will be able to revise faster. Maybe you will gain about half an hour. So another half an hour's additional material gets added to your revision. Over time, over about 6 to 8 tests, by the time you complete 6 to 8 revisions, you should be in a position to complete syllabus map, level 1 and all the perspective notes. So because with every revision, your speed will increase. All right? Stick to it. Don't deviate. All right? Give me the book. And in the interaction, few things related to the classes that I have got as feedback. One, some of you have difficulty in understanding the language of NCERT book. Not used to NCERT language. And different students have difficulties in different topics. So, over the next few sessions, today, tomorrow, Wednesday, we have 3 days. I'll be leaving on Wednesday. And in the next cycle also, I want to let it, the topics to be decided by you, not chapters. Don't tell me chapters. Come up with topics that you are struggling. Whichever topic I hear, anybody can say, you can tell this is the topic that I am struggling with, teach that. So we'll go that way, topic by topic. And I will use NCERT as the basis of that. Quickly read from NCERT and expand that. So you will get to understand from the NCERT perspective, and because I will be dealing with it, I will be expanding it a bit out of NCERT also. All right? Would that be okay? We'll do that. This is 12th. We'll complete biotechnology, the last part. You can take out your book also now. Think of the topics that you have problem with. So we have discussed all the entire process of genetic engineering. We will read 9.3. 9.3, processes of recombinant DNA technology. That is the summary of the entire chapter. All the concepts that we have discussed is summarized there. Recombinant DNA technology involves several steps in specific sequences, such as isolation of DNA, fragmentation of DNA by restriction endonuclease, isolation of a desired DNA fragment, ligation of the DNA fragment into a vector, transferring the recombinant DNA into the host, culturing the host cells in a medium at large scale, and extraction of the desired product. What happens in each of these steps? Can you recall what are the key things that you have learnt in each of these steps? How do you isolate a DNA? How do you know where the location of the DNA is? By the understanding of the locus. If we already know where the locus of a particular gene is, we will identify that locus, or we will identify by the sequences that we know of. There is a project that has been done. It is a multinational project. That is multiple countries collaborated on this project to understand the human genome. Human Genome Project. You are aware of it? Human Genome Project. How many genes did we identify in the Human Genome Project? About 20,000 genes. About 20,000 genes in the human genome were studied. And all of these genes were part of less than 5% of the DNA that we had. Alright. So we have understood about 5% of the DNA in the entire DNA sequences that we have studied. And this constitute about 20,000 genes that we have already studied. Is that all that is functional in human body? Probably not. What we have identified is what we have identified. What we don't know is 95% of the DNA. That includes all the junk DNA. What do we call as junk DNA is DNA which is non-functional at this point of time. What we don't use now. Don't use in the sense which doesn't become useful for today's survival. Then why is it there? If a part of the DNA is not being useful for today's survival, why is it there? Less than 5% of the DNA is functional DNA. More than 95% of the DNA that we have in our cells is apparently junk DNA. Why do we have junk DNA then? Why is it there? Why do we have junk DNA? What does that mean? The transmission of genetic material has been happening since the time life has been formed on this planet, correct? So according to the origin of theory, the origin of life theories, life originated about 3.5 billion years ago. Since 3.5 billion years, DNA is being transferred from one generation to the other generation. With time, more and more genetic material getting added. What is useful at a particular point of time is being functional. Rest of it is remaining there. You are not chopping it off, no? You are carrying it forward. It is like carrying all the old items from generations. Everything is being carried forward, but what you use is what is useful for you. So that is identification, isolation of DNA. Fragmentation of DNA is by restriction in the nucleus. What class of restriction in the nucleus do we use? Class 2. What is the speciality of class 2 restriction in the nucleus? Two important features. It will create sticky ends, not blunt ends. And the second one is, it does not methylate. It doesn't do methylation of the cut ends. And the third factor is, the sites of incision or the sites of cleavage where it cuts is consistent. It doesn't cut randomly at different places. It cuts more or less at the same location at all times. Okay, those are the three factors. Isolation of the genetic material. Recall that nucleic acid is the genetic material of all organisms without exception. In majority of organisms, this is DNA. In order to cut DNA with restriction enzymes, it needs to be in the pure form. So how do we purify DNA from a cell mixture? You have cells, you don't get DNA. You take cells from an organism. To extract DNA alone from that, what do? You treat that sample that you have taken with all the enzymes that can degrade every biomolecule except DNA. So you use enzymes, you use amylase to digest all the carbohydrates, proteases to digest all the proteins, lipases to digest all the fat, to break down all the membranes, ribonucleases to destroy all the RNA. You don't use deoxyribonuclease. That means when you treat a cell mixture with a when you treat a cell mass with a mixture of all of these enzymes, every biomolecule will get fragmented into monomeric form. Every biomolecule is destroyed except DNA, which will be in the polymeric form. Now to isolate this DNA, what do you add? Chilled ethanol. So you add chilled ethanol to crystallize this DNA. Then you take it. Once you take it, you cut the DNA at specific locations. How do you cut it? Restriction enzyme digestions are performed by incubating purified DNA molecules with the restriction enzymes. What do you mean by incubation? What do you mean by incubation? See, every word is important. In a regular, let's say a +2 course, going with NCERT word by word, slowly expanding on all of the things that needs to expand is the approach. But here, unfortunately, we don't have so much time. At least topic by topic we will do so that you get a hang of how to decode the NCERT. What is incubation? To incubate is to allow something to rest. To incubate is to allow something to rest in a controlled condition. Incubation of eggs. You incubate eggs. For artificial or assisted reproductive techniques, for assisted reproduction, you will incubate eggs in certain conditions which will allow the egg to survive. You will incubate embryos. Embryos are incubated, no? You collect the, by stimulating ovulation, ovulation, you collect the ovum, incubate it, keep incubating it, freezing it. Once you have about 8 to 10 ovum, once you have 8 to 10 eggs, you will bring about artificial, ICSI, intracytoplasmic sperm injection. So you bring about fertilization, allow the zygote to fertilize, allow the zygote to develop into a 16 cell stage, then you freeze it again. You are incubating it in cold temperature. You are allowing it to rest. Okay, incubation is to allow it to rest. Now what are we doing? We are allowing the DNA that has been isolated to rest along with restriction enzymes. What is the speciality of enzymes? Whatever the enzyme is, what does the enzyme do? It will act on the substrate. Whatever it is supposed to act on, it will act on that and bring about the reaction without getting used up in the process. So if you are allowing the DNA to rest along with the restriction enzymes, what will the restriction enzymes do? They will keep on looking for the sites of attachment, binding site. What is the binding site for these restriction enzymes? Where do they bind? They bind to specific. What do they bind to? They bind to specific palindromic sequences. When they bind, they cut. So when you give time for the restriction enzymes to act on the DNA is chopped off into multiple pieces wherever the restriction enzyme identified its binding site, binding site. All right. That is the restriction site. How many restriction enzymes can you use to incubate it along with the DNA in this process? Options are 1, 2, 3, 4. How many can you use in this process? How many restriction enzymes can you use in this process? One only. You don't want multiple recognition. You will use only one restriction enzyme. And the same enzyme is used to treat the vector. Vector is plasmid. All right. Agarose gel electrophoresis is employed to check the progression of a restriction enzyme digestion. DNA is a negatively charged molecule, hence it moves towards the positive electrode anode. This was another confusion that I used to have earlier. Which one is positive, anode or cathode? So many times it leads to confusion. So DNA moves towards anode or cathode. DNA is a negatively charged molecule. Why? What gives negative charge to the DNA? Huh? What gives it? See, there are two charged components in DNA. There are three parts of DNA. Nucleotide, there are three things: phosphate, sugar, and nitrogenous base. Which one is negatively charged in all these three? Phosphate is negatively charged or positively charged? Phosphate is positive. Huh? Phosphate is negatively charged. Nitrogenous base is positively charged. Phosphate is acidic. Nitrogenous base is alkaline. The acidity provided by the phosphate group is higher than the alkalinity of the base. That is why nucleotide is acidic in nature. Nucleic acid you call it. Because it has both acidic as well as alkaline properties, what do you call this kind of a substance, chemical? Amphoteric molecule. Nucleotide is an amphoteric molecule because it has a both positive and negative charge. Phosphate group is negatively charged and nitrogenous base is positively charged. It is a, what do you call something which has dual charge, both positive charge and negative charge? Zwitterion. So nucleotide is amphoteric and zwitterionic in property. Because it is negatively charged, net negative charge on this thing, when you run electrophoresis, it will move towards positive electrode. Positive electrode is anode. So eventually, I correlated cultural prejudice to electrodes. Cultural prejudice is cat is considered to be a bad omen, negative. Correct, no? Cat is a bad omen. You are driving and suddenly cat crosses your path. More or less, most of the people will slow down. Whether they stop or whether nobody or very few people would just feel like, okay, they continue at the same speed. They will at least slow down and then pick up. So cat is considered to be a negative omen, bad omen. Cathode is negative. That's all. So cation is positively charged or negatively charged? Why? Cation is positively charged. So a cation is something which will move towards, cation moves towards? Again, I'm still confused. You tell me, cation is negatively charged or anion is negatively charged? Anion, huh? So this confusion is still persisting. You please refer to your chemistry. I only know about the electrons. All right. So DNA will move towards anode. Process is repeated with the vector also, vector DNA also. The joining of DNA involves several processes. After having cut the source DNA as well as the vector DNA with a specific restriction enzyme, the cutout gene of interest from the source DNA and the cut vector with space are mixed and ligase is added. See, we have treated the vector with restriction enzyme. We don't know which vector is actually being cut or not. Then how do we select the vector which has been cut out with the space in between? It is not enough to run electrophoresis for the DNA fragments. When you run electrophoresis with the DNA fragments get separated according to its size. Correct? You know what is the size, what should be the size of your DNA fragment of interest. Whatever you are interested in, you know what should be the size. So you know exactly by calculation, mathematical calculations, you know where that particular fragment should be found on the gel. Yes or no? You know the length of the electrophoretic gel, agarose gel. You know the electric charge that you are applying for electrophoresis. You can calculate mathematically what distance will be traversed by the DNA fragments of what size. And because you already have calculated what should be the size of the fragment that you are interested in, you know where to expect the fragment on the DNA, on the agarose gel block. Correct? Vector also is treated with the same enzyme. You treat the vector, the plasmid, also with the same restriction enzyme. That means the circular plasmid is cut at two places, creating two fragments of the plasmid. One fragment is what we have to discard. The other fragment, the bigger one, is the vector with cut ends. So you run the plasmid also on electrophoresis to separate out the fragments. You identify where your vector of interest is, take that. You take that, you take your DNA of interest, again put them in a common medium, allow them to rest in the medium so that the complementary sequences will align with each other. They form a weak hydrogen bond. The backbone is not sealed, but the bases will come together. Because of the sticky ends, the complementary bases will start pairing. Now when you allow this mixture to rest for a while, you have increased the chances of the vector and the gene of interest coming with each other and form loose, weak hydrogen bonds. Then you add what? You add the molecular glue. What is the molecular glue? Enzyme? Ligase. Enzyme ligase. Ligase enzyme will bind the backbone. Wherever there is break in the sugar phosphate backbone, it will create that bonding. So now the DNA is completed. That is the process. Amplification of gene of interest using PCR. PCR stands for polymerase chain reaction. In this reaction, multiple copies of the gene or DNA of interest is synthesized in vivo using two sets of primers, small chemically synthesized oligonucleotides that are complementary to the regions of DNA, and the enzyme DNA polymerase. The enzyme extends the primers using the nucleotides provided in the reaction and the genomic DNA as template. If the process of replication of DNA is repeated many times, the segment of DNA can be amplified to approximately billion times, that is 1 billion copies are made. Such repeated amplification is achieved by the use of a thermostable DNA polymerase isolated from a bacterium Thermus aquaticus, which remains active during the high temperature induced denaturation of double-stranded DNA. The amplified fragment is, if desired, can now be used to ligate with a vector for further cloning. I should not be wasting time in the reading process. No? See, here again, there is a mistake that they have created in the sequence. In the previous segment that we studied, cloning of DNA at specific locations, they are already talking about the vector, joining this DNA with the vector. Now we are talking about amplification of DNA by using PCR. Are you amplifying the hybrid DNA or the recombinant DNA, or are you amplifying the gene of interest? What are you amplifying? You are amplifying the gene of interest. But in the book, they have given it after they have already joined it by using ligase. Correct, no? It is the sequence that is being given. When you cut it, you cut that, you cut this, and you join both using ligase. Fair to mention that. But if it is not mentioned that this will be done after the joining will be done after we amplify the gene of interest, confusion will come when you read PCR after the joining has been done. So it is not amplification of the recombinant DNA, it is amplification of the gene of interest. PCR, polymerase chain reaction. Important. There are three terms there. Reaction means what? Something which can be done repeatedly. Chain of some fragment. Polymerase is something which will create a polymer. DNA is what? Is DNA a monomer or a polymer? The DNA is a polymer. Now, we have cut out, this is the DNA fragment that we have taken. This is the DNA fragment of our interest. This is the DNA fragment of our interest. We want to amplify this. We want to amplify this and increase its copies, the number of copies that we want to increase. So what do? We will create primers. You have to break this You have to break this. See again, you break this, you cannot duplicate this before you separate this. During the natural DNA replication process, what happens? You have helicase enzyme, which will break the double helix. You have unwindase, which will uncoil the DNA. Uncoil the DNA, helicase will break it. Then you have polymerase, which will start building the complementary sequence. But you don't have helicase in an artificial medium now. So what do? You take the DNA of interest, you heat it. Heating will do what to the DNA? Heating will break the hydrogen bonds. Hydrogen bonds are as such weak bonds. When you heat it, the hydrogen bonds are broken, making the DNA into single strands, making the DNA single stranded. Now, you know what is this end of the DNA and also this end of the DNA. You know what is the what are the ends of the DNA. Now, the gene of interest that we have isolated has sticky ends. Remember? It has sticky ends. If you polymerize it and create multiple copies, you have to retain the sticky ends. Yes or no? You have to retain the sticky ends. See, DNA polymerase has a characteristic. In the DNA replication process, do you remember the initiation of DNA replication happens by the DNA dependent RNA polymerase? Do you remember that? DNA dependent RNA polymerase. So this DNA dependent RNA polymerase will create the initial segment. What is that called, initial segment? That is called the primer there also. Even in DNA replication, you call that a primer. Only when there is a primer, DNA polymerase can add more nucleotides to that. DNA polymerase cannot start the replication process. DNA polymerase cannot start the replication process. Understand this. PCR, understanding of PCR is very, very important. So, I'll slow it down because I want you to have your... Just pay attention for 2 minutes. We will understand the basics of DNA replication. That should be understood to understand PCR. Got it? See, if I read from NCERT and explain or do like that only, it will still be going from somewhere to everywhere and come back to that somewhere. I can't help it. I can't have loose ends in understanding when I explain something. All right. So this is strand A, strand B. Helicase opens it, correct? This is the replication fork. This is the replication fork. You have one is 3' to 5' and the other one is 5' to 3'. 5' to 3'. Nucleotide. See, at the 3' end, what do you have? OH group or phosphate group? Nucleotide structure. Huh? At the 3' end, you have OH group. At the 5' end, you have phosphate group. The replication progresses 3 to 5 or 5 to 3? The replication progresses from 3 to 5 or 5 to 3? 3' to 5' or 5' to 3'? So attachment of new nucleotides happen with the free OH group. Free OH group to which the hydroxyl group is attached. Alright. So now A, T, C, G nucleotides you have. You have predetermined primers. You have predetermined primers and you have polymerase. What polymerase? TAC polymerase. Thermus aquaticus polymerase, bacteria polymerase. Why this polymerase? It is heat stable. The regular human DNA, human cell polymerase or the eukaryotic cell polymerases are thermolabile. Thermolabile means what? You heat it, it will get denatured. This fellow can withstand high temperature. Now, we have double-stranded DNA. These are all there. If you put DNA into this, if there are open ends, in the DNA, what will happen? What will happen when there are open ends and there are nucleotides? The nucleotides will go and find the open ends. The complementary nucleotides will go and bind to the open ends, no? To the sticky ends, the complementary nucleotides will go and attach. Yes or no? The complementary nucleotides will go and attach to the sticky ends. So, as soon as you put your gene of interest into this medium, your sticky end becomes closed. Can we afford that? We want that to remain as a sticky end. We will see how to do that. Alright? But fortunately, the frame will not get sealed. It will only go and bind by hydrogen bonding. The nucleotides will go and attach to the complementary nucleotides on the DNA strand by hydrogen bonding, which is weak. You can easily remove them. Correct? You can easily remove them by simple heat treatment. Alright, understand? Now, this is the gene of interest. So what do we have here? This is our sticky end. This is our sticky end, correct? When we replicate this, when we make copies of this, even after that, even if we make a billion copies, we want all the billion copies to be like this only. We don't want nucleotides to be added here. We don't want nucleotides to be added here. Yes or no? Otherwise, this entire gene of interest will become useless. How do we solve this problem? Now we know in the PCR chamber to create copies, we have to add nucleotides. When nucleotides are added, nucleotides by their property will go and bind complementarily to the available bases. So nucleotides will go and attach here. And we want to keep this sticky end. We are in a sticky situation now. How do we get out of it? Replication, you cannot create copies of this without having the action of, you cannot create multiple copies of the DNA without the action of polymerase. Now, polymerase action requires the presence of primer. Primer is a short segment of nucleotides, some 8 to 10 nucleotide long DNA sequence. And that primer also can go and bind to the wherever the fragment is, wherever the primer has a complementary sequence, the primer also can go and bind by nitrogen bonding. But it will be stronger than individual nucleotides bonding because it is a 8 to 10 nucleotide long strand. It is a long strand that is attaching. It is not individual nucleotides that is bonding by hydrogen bonding. Now, how do we do it? We will not, it's a pre-decided primer. It is a pre-decided primer. Now, semi-conservative mode of replication. So, we need to have primer from this end also. We need to have a primer from this end also. And we are not creating primer as a complementary sequence to the sticky end. If we create primer to the complementary as a complementary sequence to the sticky ends, then the primer will be the same. Because sticky ends are it is the same from both ends, it is the same. We don't create primer to the sticky end. We create primer here to this. You understand? So primers, you know this is which gene it is. You know what is the sequence here. You know what is the sequence here on the opposite strand. So you create a complementary sequence to this 8 to 10 nucleotides and a complementary sequence to about 10 nucleotides on this side. You load up the medium with primers. Now primer is ready. When the DNA is made single-stranded, the primer will go and attach here. This primer will go and attach here. Primers are single-stranded or double-stranded? Primers are single-stranded. Single-stranded. Are you understanding? So now what do? This much understanding is enough. Now you heat the medium, you add DNA into this, you heat it, hydrogen bonds will break, DNA will become single stranded. As you start cooling it down, primer will go and attach. Once there is a primer available, DNA polymerase will go catch hold of the nucleotides and complete this replication. Cycle 1 of polymerase chain reaction is complete. You doubled the content of the gene of interest. Next, what do? Repeat, repeat, repeat. Keep repeating it until you have sufficiently amplified quantity of gene of interest. Is this clear? This is all polymerase chain reaction is. That's all. So, for a simple way of telling it, what is polymerase chain reaction? Polymerase chain reaction is a technique of amplifying a gene of interest, amplifying a fragment of DNA. Whether it is a gene or anything, you can amplify it. What do you need? You need the DNA fragment that needs to be amplified. You need ready-made nucleotides, all four, A, T, C, G, all the four nucleotides in ready-made nucleotides in adequate quantities. You need the initiation sequences, complementary sequences. What do you call the initiation complementary sequences? Single-stranded primers. And you need to have the Taq polymerase, DNA polymerase from bacterium, Thermus aquaticus. Now when you heat the medium, when you heat the DNA, the hydrogen bonds are denatured and the DNA becomes single-stranded. As you start cooling it, the primers will go and attach to its complementary sequences, initiating the DNA polymerase action. DNA polymerase will come into action and start elongating the DNA strand as a complementary sequence to the single strand. Now once the DNA polymerase completes the traversing of the entire stretch of the single stranded DNA, 2 copies of the double stranded DNA is produced. Doubling of DNA happened. You want to double it further? Repeat the process. Heat, denaturation, primer attachment, polymerase action, completion. Cool it down. Repeat, repeat, repeat. All right. So now by this you have created any number of you can create any number of copies of the fragment of DNA of your interest. In this case, for our genetic engineering, it is the gene of interest that we amplified. Is that clear? Did we retain our sticky ends also? We are not attaching the primer here. So after duplication also, it will still hold on to the sticky end. It will still retain the sticky end. All right? Clear? That's all. Now we will read again. PCR stands for polymerase chain reaction. In this reaction, multiple copies of the gene of interest is synthesized in vitro. What is in vitro? In an artificial environment. Vitro means artificial environment, outside of the body. Inside the body means in vivo. Using 2 sets of primers. Why 2 sets of primers? One for each of the complementary strands of the DNA. Will the sequence of nucleotides on the 2 sets of primers be the same? No. The sequence of nucleotides on the 2 sets of primers will be different, and it will be complementary to the sequences of DNA that you find on either end of the gene of interest. So what is primer? Small chemically synthesized oligonucleotides. Oligonucleotides means what? Oligo means a short chain. Something not single but not very long. Polynucleotides is long. How many nucleotides should we consider as oligonucleotide? 2 to 10, Okay, whatever, small length. There is no rule as such on considering only up to 20 you consider oligo, 20 onwards you consider poly. No. Poly is a reference to very long. Oligo is a reference to not so long. Okay. And the enzyme DNA polymerase. The enzyme extends the primers using the nucleotides. Now you know why should we have a primer? Because of the inability of the DNA polymerase to initiate DNA replication, it can only continue DNA replication when there is an available nucleotide base. When there is an available nucleotide reference for it to extend beyond that. And the genomic DNA as template. In the process of replication of DNA, if the process of replication of DNA is repeated many times, the segment of DNA can be amplified up to approximately billion times. Now, okay, you can amplify it to however number of copies you want. All right. Such repeated amplification is achieved by the use of a thermostable. See, stable and labile are opposite words. Stable means something which will withstand. Labile means something which cannot withstand. And when we use thermostable, it means that which can withstand heat. Thermolabile means that which cannot withstand heat. Isolated from bacterium, Thermus aquaticus. This bacteria is important. Questions have come on what is the source of Taq polymerase, which remains active during the high temperature induced denaturation of double-stranded DNA. The amplified fragment, if desired, can now be used to ligate with a vector for further cloning. Can we amplify the plasmid also by PCR? Can we amplify the plasmid also by PCR? Can we or can we not? We can. Doesn't matter whether it is plasmid or bacterial DNA or whichever DNA it is. PCR is a technique which can amplify DNA. So if you don't have enough number of copies of the vector, on one side you amplify the gene of interest, on the other side you amplify the plasmid. On both sides you are keeping the sticky ends intact, and then you bring them together into a common medium. Insertion of recombinant DNA into the host cell or organism. There are several methods of introducing the ligated DNA. What do we call the ligated DNA? Recombinant DNA or genetically engineered DNA. Into recipient cells. Recipient cells, after making them competent to receive, take up DNA present in its surroundings. What is making the recipient cells competent to receive the DNA? Transformation can be induced. All right. Now, you want as many host cells to take up the recombinant DNA as possible. Use all possible ways to induce the cells to take up. One, you add certain chemicals into the medium which will make the pores on the membrane of the host cell bigger, allowing the DNA to be taken up more successfully. So you are inducing transformation of bacteria. Second, using biolistics, gene gun. All right, we'll see. So if a recombinant DNA bearing gene for resistance to an antibiotic, example ampicillin, is transferred into E. coli cells, the host cells become transformed into ampicillin resistant cells. If we spread the transformed cells on agar plates containing ampicillin, only transformants will grow, untransformed recipient cells will die. Since due to ampicillin resistance gene, one is able to select a transformed cell in the presence of ampicillin. The ampicillin resistance gene in this case is called a selectable marker. Selectable marker. Now, there are two ways of looking at it. With selectable marker, there is something. The selectable marker can be the fragment of the gene into which we have introduced our gene of interest. Or selectable marker can be something else which will be used as a marker for us to identify. Now, consider this plasmid. This is the segment into which we have introduced the gene of interest. All right. This is the gene of interest. Now let's say this plasmid also has another segment, ampicillin resistance gene. This plasmid has another ampicillin resistance gene. We have introduced our gene of interest into another segment, whichever segment we have introduced it. Now any bacteria which takes this up, any bacteria which takes the recombinant DNA will be resistant to ampicillin, correct? Bacteria which does not take this plasmid will not be resistant to ampicillin. Now what if the bacteria is already resistant to ampicillin, the host? What if the host is already resistant to ampicillin? Now, we have a host bacteria. We multiply this bacteria. We have, let's say, 1 billion bacteria in the medium. We have made billions of copies of this plasmid. We put this plasmid into the medium. We induce the transformation. We push them in. We try to fire this plasmid to the bacteria, making them get transformed with this plasmid, and all that we do. Now, if this plasmid has been taken up, this plasmid will give the bacteria ampicillin resistance. This is a selectable marker. That means which will help us identify whether the bacteria has taken this or not. Correct? Now, you add ampicillin into the medium. Whichever bacteria can still multiply in the presence of ampicillin has ampicillin resistance. So we come to a conclusion that the bacteria which has ampicillin resistance has probably taken up our plasmid of interest. It has taken up the vector that has our gene of interest. Correct? How do we know the bacteria which we have selected is not ampicillin resistant already? If it is already ampicillin resistant, then in the presence of ampicillin, it will still grow. We will not be able to make out whether it has taken up this plasmid or not. How do we know the host that we have selected is not resistant to ampicillin? And if at all ampicillin resistance will come, it will come only because of our plasmid. How do we know this bacteria is not ampicillin resistant already? Or what if this is a mixture of bacteria, some of which are ampicillin resistant, some of which are not? We don't know, no, correct? We don't know which one is resistant, which one is not. We cannot check individually. So how do you test for the presence or absence of a particular antibiotic resistance in a bacterium? How do we check it? We need to find out. No, we are dealing with microorganisms whose sizes are in the span of microns. How do we figure out? Give me some idea. That is later. How do we know whether it is already not resistant? How do you ensure that you are selecting ampicillin sensitive bacteria for transformation? So you have a stock of bacteria. You have a stock of bacteria in a medium. How do you know where there is bacteria in the medium? In a culture medium, presence of bacteria can be seen as clusters, turbidity on the medium. The turbid spots on the medium are the colonies of bacteria. Each colony represents one species of bacteria. You will not have multiple species of bacteria in the same colony. Alright, that is the characteristic of that. If you have other species bacteria in this colony, they will die. So they tend to come together and grow together, the same species. So you have multiple spots of bacterial growth on the medium, whatever medium you are using, nutrient medium. Now into this medium, add ampicillin. Add ampicillin in small doses, which can, slow down metabolism and which can make the sensitive bacteria inactive, but not kill them entirely. So what is this? Sensitizing dose of ampicillin you will add, and you will make the new medium very rich in nutrients. So if there is resistance in a particular colony, that colony will not be affected by the presence of ampicillin. Yes or no? If there is already resistance to ampicillin in that colony, they will not be affected by ampicillin's presence. They will continue to thrive. The colony will continue to grow in size. That colony whose growth will stop or whose size will reduce because of ampicillin. Some of them might die also, no, if they are sensitive. Some bacteria might die and their growth is arrested, so they are not growing in spite of being in a nutrient-rich medium. So the colony shrinks or the colony does not grow. Then you know that is the colony which has sensitivity to ampicillin. So do you want to select for the next process for recombinant DNA insertion Do you want to select the colony which is thriving or the colony which is shrinking? Which one do you want to select? In this example that I gave, you want to select the colony which is shrinking, which is going down, because that is the one which is sensitive to ampicillin, and now that is the one into which we are giving this new plasmid with our gene also, and also ampicillin resistance. Now when we transfer this transformed recombinant, now think about this. You bring this colony which was shrinking into another medium without ampicillin. You take them, you put it into another nutrient medium. What will they do? They will start again multiplying and thriving. Then you bring in the plasmid, you bring in the vector, put it into the medium, induced transformation, allow them to take up the plasmid. Now there is another problem. Some of them might have taken up the plasmid, some of them might not have taken up the plasmid. We are not sure which one has taken up the plasmid. Correct? We are not sure about that. We want only those bacteria for the next stage which has taken up the plasmid, so that we can make them grow, and when we stimulate, they will produce our gene of interest product. Otherwise, if you have bacteria which does not produce, unproductive bacteria in a culture medium, which is specifically meant for producing a product, will eat away your resources and reduce the productivity. Correct? No? We don't want reduced productivity. When we put this bacteria into a culture medium and start growing them and multiplying them, and in the downstreaming process, we want insulin. So we want only insulin gene containing bacteria to be present there. So for that, we have to select them. How do we select? The selectable marker will help. Now the selectable marker in this case is ampicillin resistance. So once you complete the process of inducing the plasmid entry and all that, you treat this medium with, what do you treat it with? Ampicillin. You treat the medium with ampicillin. Now, whichever colony is thriving is the one which has been transformed. The colony which will decline is the one which has not transformed. Ignore the one which has declined, pick up the ones which are thriving, and those are the bacteria which are our hosts. First you selected the one which declined before transformation. After transformation, you are selecting the one which is thriving. Alright, time to time. So it's like life, no? You are going through so many difficulties, so many challenges. You keep cursing God. God, why are you giving so many difficulties, so many challenges? Look at him, he is thriving in the same condition. I am not able to progress. You don't know who will be selected for the next stage. Alright? A thriving colony did not get selected. A declining colony got selected. Because what is useful for the next stage in which process is understood by the one who is dealing with this process, not by the bacteria. Bacteria does not know. It can only experience what it is going through. It does not know what it is being prepared for. Correct, no? So every time you write something related to God on the board, that is what is triggered in my mind. You are being prepared for something. What is that? Nobody knows. We will not know. Somebody is preparing you for something. If you are sincere in that preparation and submit yourself to everything that you get exposed to in life, without resistance, you are being given a lot of difficulties. Without cribbing about it, running away from it, face it and go through it and become stronger. You went through that and you developed some abilities. Who knows where that ability will be used? What that ability is for? You don't know the entire game. You are just a pawn in the game. Alright? How many dimensions do you have? Your existence is how many dimensions? 3 dimensions? How many dimensions does your shadow has? 2 dimensions? If you look at it from the point of view of the shadow, shadow's existence is 2-dimensional. Shadow will think it is acting by itself. Shadow moves, shadow acts, shadow fights also. If two people are fighting here in 3 dimension, shadows are fighting in 2 dimensions. Correct? If a 2-dimensional shadow is a reflection of a 3-dimensional existence, by the same logic, cannot be a 3-dimensional existence be a shadow of a 4-dimensional existence? Can it not be? Can we deny the possibility of that? What if we are 3-dimensional pieces of a 4-dimensional game? So just play along. You don't know what you are going through is for what. To go through or not to go through, to willingly grow through it. As you go through, if you grow through it, then you are ready for the next level of the game. In any video game, in any computer-based game, as you go to higher levels, does the difficulty increase or decrease? Increase. So, you are thinking the preparation to crack NEET and to get into a medical college is a very difficult challenge. Once I do this, you are being told also, no? People will tell you, only this is the most difficult part. Once you are done with this and you get into a medical college, life becomes super easy. You can enjoy after that. All right? That is why whenever I design anything, I will make sure I design enjoyment also as a part of it. There is nothing enjoyment after that. It should be a part of your day-to-day. All right, you should play every day. You should have your fun. You should have your happiness every day, along with your focus. All right. So don't be under the illusion that the next level of the game will be easy. No. All right. It is just the trick of people to push you into the game. All right. Every stage they will tell that. 10th standard also same thing was told, no? Wasn't it told in 10th standard? 10th standard is very, very important. You have to go through this. No? 10th standard is most important. And once you are done, life is easy. You can enjoy. 12th is very important. Do that. Then you can enjoy. All right. All right. So, is this clear now? Say, you insert insulin gene into the lac operon functional gene part. Into the lactose functional gene part. Lac operon functional gene part. So what will activate that lactose producing lac operon gene? The presence of lactose. If you add lactose in the medium, that will trigger the production of all the 3 functional genes. Translation of all the 3 functional genes. What are the 3 functional genes in lac operon? ZYA. What do they produce? Beta galactosidase, galactose permease, and galactose transacetylase. Those are the 3 enzymes that ZYA produce. So you might have taken out this ZY component and put your insulin gene. The presence of lactose will activate the promoter segment of the lac operon. Operator gets activated. It will start transcribing the ZYA components. But now you don't have the galactose metabolism genes there. You have the insulin gene. Operand does not understand all that. It will just run through it, produce the insulin protein, and it is being released. Because you are not producing enzymes that will metabolize lactose. Lac operon is for what? If there is lactose in the medium, lactose is a source of energy for this bacteria. It will produce enzymes. Beta galactosidase will break down lactose. Galactose permease will increase the permeability of our galactose into the cell for further metabolism. Transacetylase transform this galactose into another form, acetylation into pushing it into other metabolism. So that is why it is producing these 3 enzymes. Now lactose is added. The bacteria is supposed to produce enzymes which will break down lactose, but those enzymes are not being produced. Insulin is being produced. Lactose continues to remain in the medium, continuously stimulating the gene, continuous translation of insulin gene. But in this process, whatever nutrients you have added into the medium, into the reactor, keeps getting used up by the bacteria. And the bacterial culture will start growing a lot. It will grow. So what do you need to do to keep this production running continuously? You have to keep replacing the medium. Once the medium becomes used up to a certain extent, or the bacterial growth has become so much that you are not able to use that productively, you remove a part of it, you add fresh medium. So by that what you are doing? You are removing the overcrowded bacteria along with a medium that has been used up. You are adding fresh medium with more nutrients. Then there is further growth of the bacteria, further production of insulin. So these bioreactors have made it possible for producing the gene products continuously, round the clock, continuously. And you have sensors, you have indicators to tell you when to replace the medium, what is the temperature, what is the condition, what is the level of different nutrients, what is the productivity, all of that. All right. Okay. Small volume cultures cannot yield appreciable quantities of products. To produce in large quantities, the development of bioreactors where large volumes of culture can be processed was required. Large bioreactors are about 1000 liters in capacity. Thus, bioreactors can be thought of as vessels in which raw materials are biologically converted into specific products, individual enzymes, etc., using microbial plant, animal, or human cells. A bioreactor provides the optimal conditions for achieving the desired product by providing optimum growth conditions, which is optimum growth condition is not only nutrients, temperature, pH, the substrate is the nutrients, salts, vitamins, oxygen, etc. Why vitamins? Why vitamins? Vitamins are coenzymes. Vitamins will help in enzymatic function. In a natural medium, they will have all of that. They will have all of those chemicals which will mimic the vitamins. They will be able to use that. But we have created an artificial medium. Unless you provide these vitamins also, they will not be able to, the bacteria will not be able to have optimum enzymatic function. All right. The most commonly used bioreactors are of stirring type, which are shown in figure 9.7. Stirring means continuously mixing. A stirred tank reactor is usually cylindrical or with a curved base to facilitate the mixing of the reactor contents. The stirrer facilitates even mixing and oxygen availability throughout the bioreactor. Alternatively, air can be bubbled through the reactor. Why do we bubble the air through the reactor? To keep the oxygen level optimum. If you look at the figure closely, you will see that the bioreactor has an agitator system, an oxygen delivery system, and a foam control system, a temperature control system, pH control system, and sampling ports, so that small volumes of the culture can be withdrawn periodically. We discussed that you have to remove the sample, you have to remove the contents of the reactor periodically. So you have mechanisms for all that. You have mechanisms to introduce new medium, to remove the medium, to mix the contents, to push air into it, to take samples from that to test it. All of that becomes a part of your reactor. It's like a vessel used for cooking. It's like a cooking vessel. Oh, how you have different ways of looking at how things are being cooked. Reactors also are like that, only cooking of products. Downstreaming process. What is downstreaming? Downstreaming is taking down the product of genetic engineering. Whatever you have produced with all this process, how do you take it out? That is the downstreaming process. After completion of the biosynthetic phase, the product has to be subjected through a series of processes before it is ready for marketing as a finished product. The processes include separation and purification, which are collectively referred to as downstreaming process. The product has to be formulated with suitable preservatives. Such formulation has to undergo thorough clinical trials, as in case of drugs. Strict quality control testing for each product is also required. The downstreaming processing and quality control testing vary from product to product. That's all. So downstreaming that is another range of activities that needs to be done, from extracting the product from the reactor to making it available on the shelves of the pharmacy, which is beyond the scope of our discussion to study. But all that we have to know is downstreaming is you collect the products from the reactor, purify it, process it, purify it, and package it. 3 P's. Process, purify, and package. And then you have to test it, do trials, sample, quality testing, all of that, and then you market it, sell it, and make money. All right. The right way of looking at it is you process it, purify it, package it, and make it available for solving problems. And in the process, you also make money. Same thing, 2 different ways of looking at it. Gives you different types of respect. I will use the bioreactor, manufacture insulin, process it, purify it, package it, sell it, and make money. I will use bioreactors, produce insulin, process it, purify it, package it, and solve the problem of diabetes in the world. You're still selling only, no? You'll still make money. But mindset makes such a big difference. It's not just for the sake of marketing, not just for the sake of making people believe that you have good intentions. But if your intention of producing insulin is to solve the problem of diabetes, you will make money anyways. You will make sure that the product is of utmost quality. You will make sure that there are no contaminants. You will take your trials very seriously. You will take your quality control very seriously. If a batch of your product turns out to be not so effective, you will not hesitate to discard the batch. But with the first mindset, I will process it, package it, I'll process it, purify it, package it, and sell and make money. If a batch is not so effective, if it is less effective, but you know it will not kill, with the money-making mindset, you will still push it into the market. Correct? But with the mindset of solving problems, it will not be acceptable for you to push anything of even the slightest of lesser quality into the market. Have you seen restaurants fast food restaurants mostly, the food that has been prepared in the morning is reheated and reprocessed for selling it in the afternoon? They do that. In Bangalore, you find restaurants, dosa is sold everywhere. Masala dosa is sold everywhere. In the morning, you sell masala dosa. You make that potato sabji for that. Palya, we call it. By afternoon, it will start becoming stale. The bad restaurants, which do not have focus on quality, they will make bondas out of it. You know what is a bonda? No. You take that potato, mashed potato and onion and all that balls, put it into gram flour, batter, put it in oil, fry it, and you eat it. It will be very tasty. But you are supposed to make it fresh, not use the dying potato palya and make bondas out of it and sell it. Alright? Mindset. If you are not foolishly selling things for free, you will still make money. But why do you want to do it? You need to have the right answers for that. You need to have answers which are bigger than your own profitability. Okay? When you become doctors, you will make money, good money. But why do you want to become a doctor? You should genuinely feel good when people come out of their suffering. That should give you the kick, not the money. And you will not be definitely working for free. You should have that self-respect. You should demand what you feel like from the hospital. But when it comes to patient care, you will be sincere with it. Okay, now next, which topic? Come on. Whichever topic I hear first, we'll discuss that.
Light reaction. Light reaction of photosynthesis.
Light reaction of photosynthesis. Okay, give me the 11th standard book. Light reaction of photosynthesis. Light reaction of photosynthesis is similar to our electron transport chain. The principles are the same. But here we have photosystems, which makes it a little bit complicated. But in electron transport chain also we have complexes. Complex 1, 2, 3, 4. Okay. Can you recall the principles of electron transport chain? Before I read NCERT and get into that, just tell me the principles of electron transport chain. What all comes to your mind? We discussed that earlier. What all comes to your mind when I say electron transport chain? What?
4 complexes.
There are 4 complexes. So we are talking about electron transport chain in the mitochondria. There are 4 complexes. Then go on. Whatever comes to your mind. Need not be connected to each other. Something related to electron transport chain, just keep shooting. ATP is produced. Excellent. Then
Movement of protons.
movement of protons. Where? From? Movement of protons across some membrane. Correct? So by that what we are creating? We are creating a proton gradient. So there is movement of protons and there is a proton gradient created. Then what else comes to your mind? Come on.
Transport of electrons.
Transport of electrons. So when we talk of transport of electrons, anything else that comes to your mind? You should be kind to people with physical disabilities. All right. And the fact that all of you are laughing knows that, means that you know that I am a person with physical disability. Hearing problem. Of my own making, because of shouting too much for too long. Whatever is the cause, but I can't hear if you speak low. What comes to your mind if we talk about electron transport? Ubiquinone, okay. Cytochrome C. Now you are going into the complex. Electron transporters, okay. Then go on. Come on, okay. Ubiquinone, cytochrome C. What comes to your mind when we talk of electron transport? Electron should come from somewhere. Make it more generic. No, don't make it specific to the compounds. Make it more generic about electron transport. Okay, FADH, NADH, NADP, FADH2, all of that. Okay. Then? Energy-rich molecules. Okay. Some molecules which carry energy, which can be transformed. Okay. Then? What else?
Oxygen, water.
Oxygen utilized, water being produced, metabolic water production. When we talk of electron transport chain in the mitochondria, then See, as a solution to stay awake, I told many of you that keep drinking about 100 ml water every 15 minutes. 1.5 hours I have not had a drop of water. None of you had water, as I could see. Now because I took water, she started to drink. Okay, drink some water. We'll cheers. Very important, we should remember to drink water. We don't realize that not drinking water periodically is the reason for a lot of attention problems. Okay. What else? Come on. 2 crucial things that you have left out. For drinking water is fine, okay. Splitting water, okay. Splitting water in photosynthesis, in the light dependent reaction. I thought you told drinking water. Oxidation reduction, very important. Electron transport happens with continuous oxidation and reduction of successive compounds. Then, Okay, that is specific. Some complex 2 doesn't pump proton. Okay. Come on. For electron transport to happen, you need two very crucial things. Photosystems, okay.
Production of energies. Production of energy.
Production of energy. Let's say I am a porter. I am a delivery guy. My job is to deliver this book. Two things are required for me to do this. What are the two things? Two things should happen for me to be able to deliver this book. I am the one who is transporting. Address is there. Address is there.
Electron, proton.
Talk about this book now. Talk about this book. My job is to transport this book. Without two things happening, I will not be able to transport this book. What are the two things?
Mode of transport.
I am the mode of transport.
And
Who will I give it to? Somebody to receive the book. Then one more. One more. Somebody should give me the book, no? So for transportation to happen, the transporter should receive the material and deliver the material. The pickup and drop off. For electron transport chain, what do you need for electron transport to happen? Two crucial things. Without that, electron transport cannot happen. You need electron donors and you need electron acceptors. That's all, no? Common sense. Okay. So these are the basic principles. Somebody should give electron. An electron should be excited. Electron should be transported. Excited electron is capturing energy. Excited electron has high energy. As electron moves from one to the other, to the other, to the other, by the time it reaches the end receiver, it is falling down in the energy state. Yes or no? As electron moves from one whatever it is, one substance to another substance, what will happen to the state of energy or the amount of energy that the electron holds? When it moves from A to B, the amount of energy that the electron holds when it reaches B, will it be more or less or same as it was when it was at A? It will be less. That means when electrons move from one substance to another substance to another substance in the chain of electron transport, it is giving out energy. It is giving out energy. Now, an effective electron transport chain should have mechanism of using this energy. For what? For doing something. We'll see what it is. But it should have some mechanism of using this energy. In mitochondria, the energy is used for what? For pushing protons outside of the matrix into the intermembrane space. Here also, in light reaction of photosynthesis also, the energy of the electron that is being lost when electron moves from one carrier to another carrier is used for pushing protons. Where? Where? From the stroma to the is it see, from in mitochondria it is inside to outside. In chloroplast it is outside to inside. From the stroma to the thylakoids. It gets pushed from the stroma to the thylakoids. So that energy is being used. Principles are the same. You create a proton gradient. The proton gradient, I'll play some song also. Ye dostati hum nahi chodenge. All right. In the middle of the class, they are happily enjoying their friendship, holding hands, dancing in the mind. Knowing both of you might probably be dancing also in the mind, happily singing. All right. Okay. Okay. So with this context, we'll look at it. Open the photosynthesis chapter. We'll directly go to the electron transport, light reaction, page number 138. See, chloroplast structure, you know, there is stroma and there is grana. Grana is a stack of thylakoids. Thylakoids are the plates there. Whatever plate-like stackings that we have, those are all grana. Let's see how it is given. Where is light? What is light reaction? 11.5. Light reaction or the photochemical phase include light absorption, water splitting, oxygen release, and the formation of high energy chemical intermediates ATP and NADPH. ATP and NADPH. Now, before going any further, knowing the purpose, knowing how electron transport happens and all that, tell me what would be the reason for light absorption. Why do you want to absorb light here? To get the electrons excited. See here, in mitochondria, how did we get the electron? We split a chemical. You broke down NADPH and FADH2. Enzymatically, you break it down, making the electron to be separated from the proton there. So you are chemically doing it. But here, this is the beginning of the initiation of organic chemical production. So you cannot get electrons by breaking down some existing chemical. You are producing the chemical. So there is nothing that is already there which you can break to release the electrons. So what else can you do to release electrons? Every cell, every atom has electrons in it. Every atom has electrons in it. There is a nucleus in the center with protons and neutrons. The nucleus will pull the electrons towards it. Closer electrons in the initial cells, in the lower shells, the electrons have high pulling force. The force that pulls them towards the nucleus, which holds them together. As you go to the outer shells, the force of proton pulling the electron will be lesser. So your chance of releasing electrons or exciting electrons out of this is higher. Correct? So where is the electron getting out from? The electron is being released by excitation from atoms. You have to somewhere excite the atoms to release electrons. How can you do that? You have a ball, you throw it up. It keeps going up and up and up and up and it goes into moon. Does it happen? Now what happens? It will fall back. But you have given energy to the ball to get out of it. Not enough to overcome the gravitational pull. So what should you do? You tie the ball to a rocket, light it up. The ball goes to moon. And if you are Elon Musk, you will also say that I am sending cricket balls to the moon so that when we go there we have enough balls to play. He is selling plots on the moon and Mars also. People are buying it. So many people struggle to sell plots on the surface of Earth. Sir, I have a site, 30-40 site. Please sell it. I'm trying to sell 30-40 site for 3 years, not able to sell. Elon Musk is selling properties on Moon and Mars. Right? That is the power of credibility. If Elon Musk is telling this, probably someday he will make it. All right? There is a Subhashita in Sanskrit. Okay? It goes like this. Swagrihe pujyate naraha, swagrame pujyate prabhu. Swarajye pujyate raja vidwan sarvatra pujyate. Meaning, every person has respect in his home. A drunkard, he is going around getting beaten up by everybody on the street. He comes home, he stands like this in front of his children. His children, oh father. Right? Everybody has respect in their homes. The village headman is respected in his village. Outside of his village, he goes to the city and says, I am the sarpanch of this village. So what? Nobody bothers about him. Swarajye pujyate raja. A king is respected in his kingdom. Let's say the chief minister of Meghalaya is respected in Meghalaya. There is protocol. In India also is respected because of part of India. Now he goes to, let's say, he goes to California and says, I am the chief minister of Meghalaya. If he goes with the diplomatic privileges, he will be still given protocol. But if he goes on a private trip and says, you follow protocol, you will not get the protocol. Vidwan sarvatra pujyate. A wise man is respected everywhere. If you are a wise person, until you start speaking, people don't know you. Once you start speaking, wherever you are, people will need you. Alright, so it is worth the effort to work and build wisdom. A knowledgeable person, a wise person is respected everywhere. It doesn't matter who you are. Your identity doesn't matter. Which country you belong to doesn't matter. How much money you have doesn't matter. You will still be respected. Okay. Why did I tell this? Electron excitation. Throwing the ball to the moon. Now with these photosystems, consider this. All right. I don't deviate from anything. I will always remain in the topic. If it appears like deviation, it will be for some purpose of linking something to something. Photosystems are like places of exciting electrons. Consider photosystems to be the places where electrons are excited. Now, excite an electron. Okay? Light harvesting complexes are the ones which will harness the power of energy in the solar or in the sunlight and gather enough energy, accumulate enough energy to excite electrons within the photosystem 1 and photosystem 2. These are named in the sequence of their discovery, not in the sequence of being operational in this photophosphorylation. In photophosphorylation, what is first operational? What is the important one? PS2, not the PS1. Got it? They are named in the sequence of their discovery and not in the sequence in which they function during the light reaction. The light harvesting complexes are made up of hundreds of pigment molecules bound to proteins. Each photosystem has all the pigments except one molecule of chlorophyll A, forming a light harvesting system, also called antenna. Antenna is for what? To absorb and transfer energy. These pigments help to make, see, chlorophyll A is where is the chlorophyll A? Chlorophyll A is in the central complex, or what do you call that? Reaction center. Reaction center, correct? Where is the electron getting excited from? Electrons are getting excited from the reaction center. So you see figure 11.4 on the same page. The light harvesting complex is given. You have multiple pigment molecules. One is mentioned as the reaction center. The reaction center is always chlorophyll A. All the other pigments are present in the antenna complex. Pigment molecules, what is mentioned, they are all part of the antenna complex. Reaction center is the one which is at the center, where you see the blue arrow being marked towards the primary acceptor. Primary acceptor is the molecule which will be receiving electrons that are excited from the reaction center. Photon is what? Photon is light. Light has dual nature. Light is a wave, light is a particle. Photon is a representation of the particle nature of light. Correct, no? If you look at light as a wave, light is referred to as wave particle, no? It is a wave as well as a particle. Photon is packets of energy that you will get in light. Those packets of energy are absorbed, because here we are talking about transforming energy, so light is being reflected or represented in the form of photons, packets of energy. Alright. So you see that photon is being mentioned, the other pigments, and there is some line indicating the connection between the antenna complexes and the reaction center. That one golden color line that they have mentioned, it is reflecting that energy from the antenna complexes are being transferred to the reaction center. Alright. Why do you have different types of pigments? See, every pigment has its optimum wavelength of absorption. The chlorophyll A has wavelength of absorption around 680 to 700 nanometers. 680 nanometer you can consider. Photosystem 1, photosystem 2 is P680, no? Photosystem 1 is P700. What is P680 and 700? Their optimum light absorption is in the wavelength of 680 nanometers. That is P680. So the light in the wavelength of 680 nanometers is absorbed by the reaction center of photosystem 2. Reaction center of photosystem 1 will absorb light of the wavelength 700 nanometer. P680, P700 is a reference to the wavelength of light that they will absorb. The light of the sunlight is not only made up of one wavelength, and we are talking about visible light, visible spectrum, 680 nanometer. What is the visible spectrum? What wavelength constitutes the visible spectrum? What? I don't remember. I'm asking you. 10 to the power of 15. Okay. Tell me in terms of nanometers. What nanometer to what nanometer is the range for visible light? 400 to 600. Then 680 nanometer is not in the visible spectrum. 380 to 760, no? Okay. Whatever, you just look at it. But here in the photosystems, we are talking about visible spectrum of the light. Why different pigments? Why different pigments? Why do we need to have antenna complexes? Why do antenna complexes have all other pigments but not chlorophyll A? Chlorophyll A is concentrated in the reaction center. Other pigments are present in the antenna complexes. The wavelength of absorption for different pigments is different. So having various pigments allows the chloroplast to be capable of absorbing light of different wavelengths. Your visible spectrum is not having only 680 nanometer or 700 nanometer. Visible spectrum has a range of wavelengths. So if you have capability of absorbing light at different wavelengths, your efficiency of photosynthesis, your efficiency of harvesting light improves, making you more productive in photosynthesis. Alright? And the amount of light absorbed by chlorophyll A alone is not enough. The other centers are connected to it so that they amplify the energy. Alright? Okay. The reaction center is, these pigments help to make photosynthesis more efficient by absorbing different wavelengths of light. The single chlorophyll A molecule forms the reaction center. The reaction center is different in both the photosystems. In photosystem 1, the reaction center, chlorophyll A, has an absorption peak at 700 nanometer, hence is called P700. Well, in PS2, it has absorption maxima at 680 nanometer and is called P680. This is clear, no? Electron transport. Now we know how the photosystems are packaged. Different pigments, chlorophyll A in the center, all other pigments are around it. So you have a spectrum of wavelength that the photosystem as such can absorb. All the energy absorbed is directed to one spot. What is the spot to which all the energy absorbed in the entire photosystem is directed to? It is directed to the reaction center. Purpose is to raise energy to such an extent that it can excite an electron. That's all. Alright? Now when you excite an electron, what are you doing to the electronegativity of the system? Because the excited electron goes away. The excited electron goes away. Oh, not electronegativity, let us look at it in a different way. You supply some energy. The energy reaches a critical point. Electron is excited. There is a primary acceptor. Whoever is the primary acceptor, we will see. All that is a matter of just listing down names of carriers. Getting the principle is important. The primary acceptor picked it up, that passed it on to something else, that passed it on to something else. Now this electron is gone. One cycle completed. To do this for another cycle, the energy required to excite one more electron, would it be the same or will it be more or less? You will need more energy to excite another electron. So you will have to harness more solar energy. That electron is also excited. The next round, third round of this process will require even more energy. So this will go on to create a system of perpetually increasing energy demand to continue to be in operation. If you want to overcome this, what do you need to do? If you want to overcome this, what do you need to do? You have to compensate the lost electron. Whatever electron is removed from this system has to be compensated. Some electron has to come from somewhere. You need to give electron to this system, PS2. Otherwise, the electron that is lost is gone. It is gone. But to excite one more electron, you will need more energy. Next cycle requires more energy. Next cycle requires more energy. The system will shut down. Where will that electron come from? Splitting of water. When you split water, you are splitting water into hydrogen ion, oxygen, and electron. The electron that is released by the splitting of water replenishes the electron lost from the reaction center. So, splitting of water is associated with which photosystem? Photosystem 2. Photosystem 2. Now, from photosystem 2 to photosystem 1, electrons are passed on. However it goes, we will see. And in the process, there are stages where you create a gradient for protons. You push protons from outside to inside. You push protons from outside of the thylakoid to inside. From the stroma to the thylakoid, you push protons. In which step does this happen is a matter of just looking at that step. But do you understand the process now? Photosystem 2 is there. It is absorbing energy of the sunlight. Accessory pigments or the antenna complexes are pushing the energy to the reaction center. Reaction center is exciting an electron. Electron is going away. Simultaneously associated with the photosystem 2, hydrolysis is also happening. There is a water splitting complex associated with photosystem 2. Is water splitting happening in the photosystem 2? Is water splitting happening in photosystem 2? No. There is a water splitting complex closely associated with photosystem 2. The electron that is released there tops up the photosystem 2 electron, which has been lost. So this completes the loop here. Electron is removed, electron is replaced. Proton is created, proton gets accumulated inside the thylakoid. There are mechanisms in the flow of electrons which will bring proton from the stroma into the thylakoid. So you are increasing the concentration of thylakoid, increasing the concentration of hydrogen ions inside the thylakoid. What will you do with these hydrogen ions? What will you do with these protons? You will run this through. You will allow passage of these protons through what? You will allow these protons to exit through what? Through ATP synthase complex. You will allow these protons to flow through ATP synthase complex. So inside the thylakoid you will fit ATP synthase complexes. The gradient is created. Any chemical would want to balance the gradient. If there is a gradient, it will flow. It will look for the conduit to pass. It will look for a passage. It wants to get out. The hydrogen ion wants to get out into the stroma because there is too much of accumulation of hydrogen ions inside. No other place will allow hydrogen ion to get out. It will be allowed only through ATP synthase. And when it flows through ATP synthase, the ATP synthase is activated to bring ADP and phosphate together, producing ATP. Clear? Also, there is more hydrogen ion. Electrons are flowing from photosystem 2 to photosystem 1. In photosystem 1, you have electron. Hydrogen ion is there. NADP is available as a raw material. NADP, hydrogen ion and electron combine together to form NADPH. So two things are being produced. NADPH is produced, ATP is produced. So whatever electrons you excited is contributing to the formation of NADPH, which is a high energy compound. NADPH production requires combining NADP and hydrogen ions. Hydrogen ions are produced by splitting of water and also by bringing in hydrogen ions from the stroma into the thylakoid. So you are creating that gradient for hydrogen ions, making it available for NADPH binding. Energy for the binding of NADPH and hydrogen ions is coming from electron, the electron. So you are not spending anything else, you are creating energy storage. You understand? And hydrogen ions are allowed to pass through ATP synthase, thereby producing ATP also. This is the mechanism. We will see the process. Mechanism, did you all understand? Did you understand the mechanism of photophosphorylation? The light reaction? Process we will see, we will read out. The electron transport. In photosystem 2, the reaction center chlorophyll A absorbs 680 nanometer wavelength of red light, causing electrons to become excited and jump into an orbit farther from the atomic nucleus. These electrons are picked up by an electron acceptor, which passes them to an electron transport system consisting of cytochromes. Okay. Figure 11.5. See, you see that light harvesting complex. Which is the light harvesting complex there? Photosystem 2 P680. From there, electron acceptor is receiving the electrons. Then through the electron transport system, it is reaching the light harvesting complex 1, that is the photosystem 1. From photosystem 1, it is being passed on to the terminal electron acceptor, which is, who is the terminal electron acceptor in this system? NADP. NADP and electron and hydrogen ion combine to produce NADPH. You see that? Associated with the photosystem 2, you see here water is being split. It is shown as a reaction, but it is a water splitting complex. It is a water splitting complex. There are multiple steps in that also, how water is being split. We will look at it as a broad thing now. So this is called Z scheme of light reaction or Z scheme of light reaction if you are more Americanized. Z. I will continue to say Z. All right. Okay. This movement of electrons is downhill in terms of an oxidation, reduction or redox potential scale. That means what? Redox potential is higher or lower as you go downhill? There should be a gradient for the flow of electrons also, no? The electronegativity gradient is in an increasing order or a decreasing order. From the beginning to the end of the electron transport chain, electronegativity increases or decreases? increase. What is electronegativity? Electronegativity is the extent of craving for electrons. The desire for electrons is electronegativity. It is the greed for electrons, the ability to receive electrons. Who should have higher electronegativity, the giver or the taker? Giver should have a higher electronegativity or the taker? Taker should have higher electronegativity because he should be able to snatch it from the giver. If the giver has higher electronegativity, he will not give it. He will hold on to it stronger than the pull that the giver is creating. So electronegativity keeps on increasing as you go down the electron transport system. The electrons are not used up as they pass through the electron transport chain, but are passed on to the pigments of photosystem 1. Simultaneously, electrons in the reaction center of photosystem 1 are also excited when they receive red light of wavelength 700 nanometer and are transferred to another acceptor molecule that has a greater redox potential. Redox potential is electronegativity only. Alright. So higher redox potential is to be present in an acceptor. These electrons then are moved downhill again, this time to a molecule of energy rich NADP. The addition of these electrons reduces NADP plus to NADPH plus H plus. This whole scheme of transfer of electrons starting from the PS2 uphill to the acceptor, down the electron transport chain to PS1, excitation of electrons, transfer to another acceptor, and finally downhill to NADP plus, reducing it to NADPH plus H plus is called the Z scheme. Due to its characteristic shape, this shape is formed when all the carriers are placed in a sequence on a redox potential scale. Alright. Now, one more knowledge point that you have to add is, just like electrons are getting excited from the reaction center of photosystem 2, electrons are getting excited from the reaction center of photosystem 1 also. They are getting excited from the reaction center of photosystem 1 also. Now this electron which gets excited from the reaction center of photosystem 1 goes to the acceptor, from there goes to the NADPH. Correct? The loss of electrons in photosystem 1 is replenished by the electrons coming from photosystem 2. But the loss of electrons from photosystem 2 has to be replenished by the electrons that comes from the splitting of water. Alright. So it has to be a zero-sum game. Zero-sum game is what you lose, you gain. At the end of it, you are in a no loss, no profit situation. But something has happened. That is this process. Right? Energy can neither be created nor destroyed. First law of thermodynamics. Okay. Right. Splitting of water. You would then ask, how does photosystem 2 supply electrons continuously? We asked it and we answered it. The electrons that were moved from photosystem 2 must be replaced. This is achieved by electrons available due to splitting of water. The splitting of water is associated with the photosystem 2. Water is split into two hydrogen ions, oxygen and electrons. This creates oxygen, one of the net products of photosynthesis. The electrons needed to replace those removed from photosystem 1 are provided by photosystem 2. That's all, no? We need to emphasize here that the water splitting complex is associated with the photosystem 2, which itself is physically located on the inner side of the membrane of the thylakoid. Then where are the protons and oxygen formed likely to be released? In the lumen or on the outside of the membrane, outer side of the membrane? Where are they released? Will they be released into the lumen or to the outer side of the membrane? Hydrogen ions. Into the lumen of thylakoids. Eventually where will they go? They will go outside of the thylakoid into the stroma. Through what? Through ATP synthase. In the process producing ATP. There are two things, no? Cyclic photophosphorylation, non-cyclic photophosphorylation. Some little bit smaller details we need to look at to understand that. We will see how it comes, where it comes. Next only it's there. Cyclic and non-cyclic photophosphorylation. Living organisms have the capability of extracting energy from oxidizable substances and store this in the form of bond energy. Special substances like ATP carry this energy in their chemical bonds. This was the first class we did. Where is the energy in living organisms? In the bonds between elements in their chemical molecules. The process through which ATP is synthesized by cells in mitochondria and chloroplasts is named phosphorylation. There are 3 types of phosphorylation. Can you recall? Phosphorylation that happens in the chloroplast is photophosphorylation, because photons are used for this. Phosphorylation that happens in mitochondria is oxidative phosphorylation. Why? It is dependent on oxygen. It is dependent on oxygen, so it is called oxidative phosphorylation. Phosphorylation or ATP synthesis happens during other chemical reactions not associated with photophosphorylation or oxidative phosphorylation is referred to as substrate level phosphorylation. Phosphorylation happening in the process of converting some substrate into some product during regular chemical reactions outside of these 2 processes. Okay. Photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate in the presence of light. When the 2 photosystems work in a series, first PS2 and then PS1, a process called non-cyclic photophosphorylation occurs. The 2 photosystems are connected through an electron transport chain as seen earlier in the Z scheme. Both ATP and NADPH are synthesized by this kind of electron flow. Non-cyclic photophosphorylation, no problem, we saw that. Electrons, water electron excited from photosystem 2, acceptor, electron transport chain, photosystem 1, electron transport, acceptor, and then goes to NADPH production. Electron flow over. Electron lost from PS2 is replaced by electron coming from splitting of water. Electron lost from PS1 is replaced by electrons coming from PS2. No problem there. Z scheme keeps going, continues to happen. What is cyclic photophosphorylation? We'll see. When only photosystem 1 is functional, the electron is circulated within the photosystem and the phosphorylation occurs due to cyclic flow of electrons. A possible location where this could be happening is in the stroma lamellae. In the stroma lamellae, while the membrane or lamellae of the grana have both photosystem 1 and photosystem 2, the stroma lamellae membranes lack photosystem 2 as well as NADP reductase enzyme. NADP reductase enzyme is the one which will combine NADP, hydrogen ion and electrons to produce NADPH. Now when you don't have that enzyme, NADPH production is not happening. So in cyclic photophosphorylation, do you have the production of NADPH? In non-cyclic, this flow happens, electrons are exiting the system. NADPH production can happen when electrons exit the system. But if electrons are circulating within the system and they are not exiting, NADPH production cannot happen. But there is a step. When cyclic photophosphorylation happens, electrons flow through the series, the reaction, where the transfer between 2 molecules, where proton pumping is happening. That means cyclic photophosphorylation will lead to proton pumping into the stroma, into the thylakoid stroma, into the thylakoid lumen, into the thylakoid lumen. Alright. So if hydrogen ion concentration is being created, but electron is not exiting the system, what energy will be produced? These hydrogen ions will exit through ATP synthase, producing ATP. Non-cyclic photophosphorylation will give rise to both ATP as well as NADPH. But cyclic photophosphorylation will give rise to only ATP production. We'll see how. So it's not given here. So I'll have to tell you the series of carriers. Right now, let us fix it that way. All right. The cyclic flow, okay, the excited electron does not pass on to NADP but is cycled back to the PS1 complex through the electron transport chain. The cyclic flow hence results only in the synthesis of ATP but not NADPH. Cyclic photophosphorylation also occurs when only light of wavelengths beyond 680 nanometer are available for excitation. One more observation you have to make. Cyclic photophosphorylation will lead to photosynthesis without the production of NADPH is fine. What else is not produced if it is cyclic photophosphorylation? Huh? Oxygen. Why? Oxygen is produced in association with photosystem 2. When photosystem 2 is not being engaged, oxygen cannot be produced because oxygen production requires the removal of electrons from the water splitting complex. When water is being split, the electrons have to be removed from the water. The phosphorylation process, it is cyclic photophosphorylation that is happening. No electrons can be pushed into the photosystem 2. That will stop or shut down the water lysing complex. That's all, no? Even if there is 680 nanometer wavelength light available, reaction center can excite an electron. But there is no acceptor to receive the electron. Then, will the reaction center excite electrons further? The photosystem 2 will shut down. What you produce needs to be taken up by somebody. If you are producing an electron, if you are exciting an electron, it has to be taken up by somebody. Then, if the conditions are still favorable, you will further excite electrons. You understand? So, half an hour now. This game should happen from school. 15-20 minutes, one glass of water. Take a shot. You have a bottle and a glass, 100 ml glass. Fill it. It's a nice break also. We also enjoy that. Everybody take a glass of water. Cheers. Children will enjoy it more. Or maybe adults enjoy it more. Children do it every day. So it's normal life for them. We do it once in a while. So an opportunity to be like children is more enjoyable to adults than being themselves for children. Correct, no? They are being themselves. We are trying to be children again. Yes. So, light reaction. What do we have here? Forget about the correctness of positioning of these complexes and all. Just look at the process. What is this? What is this? Photosystem 2. This is water lysing complex, we'll say, or water splitting complex. This is primary acceptor. So, from here to here, there are a series of electron carriers. Let's say there is somebody A, somebody B, somebody C. Alright? Now when electrons flow from A to B, consider there is alright? Protons coming inside. Protons coming inside. Now this fellow, there is something else associated here. What is this? ATP synthase. This is photosystem 1. This is acceptor again. There is somebody who is carrying electron acceptor. Electron acceptor. This is NADP, which is the terminal electron acceptor. Now this is NADPH. This is hydrogen ion. This is again hydrogen ion going out of it. This is ADP plus phosphate becomes ATP. That's all, no? That's all. For this fellow to operate, you need to create a gradient for hydrogen ions. Where is hydrogen ions where are hydrogen ions coming from? Hydrogen ions are coming from this. What is this fellow doing? This fellow is splitting water into hydrogen ion plus electron plus oxygen. So hydrogen ion is getting accumulated inside. This fellow is passing on electrons. So electron that is lost, that electrons are coming from here again. Correct? Now when these electrons pass from A to B, this fellow gives it to A, this fellow gives it to B. When electrons pass from A to B, from the stroma of the chloroplast, hydrogen ions are pumped into the lumen of thylakoid. So you are adding on hydrogen ions by splitting water. You are adding on hydrogen ions by pumping hydrogen ions during electron flow. You understand? Then electron comes to photosystem 1. This passes on electrons to something else. There are two more here. Something else will take up electrons. And hydrogen ion will be used up by this fellow also. Electrons are used by NADPH. Electrons will be received by NADPH terminally. And NADPH is produced. Final output from this photophosphorylation is you get two energy compounds. You get oxygen as the byproduct. That's all. Now these energy compounds are used during light independent reaction, Calvin cycle, to bring together carbon dioxide molecules and make glucose. So you took energy from sunlight, fixed that energy in the bonds of ATP and NADPH, used these fellows to create bonding between carbon elements, carbon atoms, to create glucose. Now energy in these high energy compounds got transformed into energy in the bond between carbon atoms of glucose. Use that glucose, break it up, load up the energy into ATP and NADPH. Use that wherever you want it in the body. See the transformation of energy? The transformation does not happen absolutely. So there is some loss of energy in the form of heat. So energy is available from sunlight. Part of it is fixed in the form of bonds in these chemicals. They are used to create bonds between carbon atoms, make glucose. Part of energy of this is lost in the form of heat. Now use glucose as a source of energy in living systems. Break it, make ATP and glucose, ATP and NADPH and other high energy compounds. Use it to join some other elements. Break, join, break, join. Ultimately you have 118 elements right now that we have identified. We know of 118 elements now. Mix and match the elements to produce different compounds. Carbohydrate to convert, you convert carbohydrate into lipids. What is carbohydrates have what? Carbon, hydrogen, oxygen. Elementally they have carbon, hydrogen, oxygen. What do lipids have? They also have carbon, hydrogen, oxygen. You arrange carbon, hydrogen, oxygen in a particular configuration, you have carbohydrate. You arrange the same carbon, hydrogen, oxygen in a different configuration, you have a lipid. You add nitrogen element into it and arrange it in a particular configuration, you have amino acids. Join them in different combinations, you have different proteins. Carbon, hydrogen, oxygen, nitrogen, all four in particular configurations gives you your nucleic acids. That's all, no? Right? Chemical transformation of material from one form to another form requires only two things. It requires the elements necessary to create that structure, and it requires the energy to create the bonding between those elements. And the third important functional requirement is you require enzymes which can bring about that configuration. Correct? Elements are the raw materials. Energy is the operational currency. Enzyme is the designer. For the creation of this, God is in the form of enzymes. That's all, no? Raw materials, energy, enzymes. That's all. This is all life is. In the physical plane. You move into the spiritual plane, life dimensions change. But in the physical plane, life is a combination of these three things. Whether it is your body, or a cockroach, or a frog, or a butterfly on the wall, plant, whatever it is, different organisms have different designers pool. That is the pool of enzymes they have. All the organisms are dealing with the same elements. What we can mix and match and use is all what plants have taken up from the soil. Elements are coming from where? Elements are coming from soil, elements are coming from the air. Other than, even carbon, oxygen, plants are taking carbon dioxide from the air. But carbon and oxygen are being taken from the soil also, no? So can we safely say our entire body is made from soil? And when we die, it again becomes a part of soil. That's all, no? We are mud. Right? So when you look at the religious way of telling about creation, God made all creation from soil, from mud He created. Different way of talking about the same things. Through evolution, through plants. So three things, the entire life is understood. Our entire study is to understand the play of these three. Elements, energy, enzymes. Done. All the entire play of life is taken care of. When we look at this, we are looking at the same thing only, no? We are looking at the play of energy to combine elements which will act as the storage of energy in the form of bonds used in a different place to create other set of combinations of elements that again mix and match continuously happening. The entire plant body is formed, we eat it, we break it down into simple structures. Our body has the enzymes to restructure the elements, create what our body needs. So whatever we eat, our body will make it into human form. The same thing eaten by a cow is made into a cow's body. A dog eats it, it becomes the dog's body. That's all. Is this clear? Electron flow, source of electrons, places of electron excitation, the source of electron replenishment, electron acceptor, proton channel. Proton channel is the ATP synthase. Outcome of this is NADPH and ATP. Done. This is non-cyclic photophosphorylation. Cyclic photophosphorylation, what happens? The electron, which is supposed to flow here, will not flow here. From here, it will go to this fellow. Now what happens? From photosystem 1, electron goes to the acceptor. From acceptor, it doesn't go to the NADPH because you don't have the enzyme NADP reductase. Electrons are diverted to this fellow, one of the electron carriers in the electron chain between photosystem 1 and photosystem 2. And when electrons come here, it will flow to B, C, and photosystem 1, this loop. You understand? So when electrons flow between these two, hydrogen ion is pumped inside. Cyclic photophosphorylation is contributing to the creation of a proton gradient. And that proton gradient will drive the ATP synthase complex to produce ATP, not NADPH. Alright. NADPH is not produced, oxygen is not produced. Alright. Chemiosmotic hypothesis. Is the conceptual clarity there now? Light reaction? Chemiosmotic hypothesis. Let us now try and understand how actually ATP is synthesized in the chloroplast. The chemiosmotic hypothesis has been put forward to explain the mechanism. Like in respiration, in photosynthesis too, ATP synthesis is linked to the development of a proton gradient across a membrane. This time, these are the membranes of thylakoids. There is one difference though. Here the proton accumulation is towards the inside of the membrane. In mitochondria, it is towards the outside of the membrane. It is in the intermembrane space. Here it is inside the thylakoid membrane. That is in the lumen. In respiration, protons accumulate in the intermembrane space of the mitochondria when electrons move through the electron transport system. Let us understand what causes the proton gradient across the membrane. We need to consider again the process that takes place during the activation of electrons and their transport to determine the steps that cause a proton gradient to develop. Since splitting of the water molecule takes place on the inner side of the membrane, the protons or hydrogen ions that are produced by the splitting of water accumulate within the lumen of the thylakoids. We saw that already. The water splitting creates accumulation of protons. As electrons move towards the photosystems, protons are transported across the membrane. This happens because the primary acceptor of electrons, which is located towards the outer side of the membrane, see, it is not outside of the membrane, outer side of the membrane. You should consider this entire thing to be the thylakoid membrane. Okay? Proton acceptor that is present towards the outer side of the membrane transfers its electrons not to an electron carrier but to a proton carrier. Hence, this molecule removes a proton from the stroma while transporting electrons. Alright? When this molecule passes on its electron to the electron carrier on the inner side of the membrane, here, the proton is released into the inner side of the lumen of the membrane. Proton is released onto the inner side of the lumen. This is the thylakoid membrane, the entire thing. This is the lumen. This is outside of the thylakoid. The NADP reductase enzyme is located on the stroma side of the membrane. Along with electrons, comes from the acceptor of electrons of photosystem 1. Protons are necessary for the reduction of NADP plus to NADPH. That is what we have seen. These protons are also removed from the stroma. Hence, within the chloroplast, protons in the stroma decrease in number, while in the lumen there is accumulation of protons. This creates a proton gradient across the thylakoid membrane as well as a measurable decrease in pH in the lumen. The lumen becomes acidic. Why are we so interested in the proton gradient? This gradient is important because it is the breakdown of this gradient that leads to the synthesis of ATP. The gradient is broken down due to the movement of protons across the membrane to the stroma through the transmembrane channel of the CF0 of the ATP synthase. The ATP synthase enzyme consists of two parts. One called the CF0 is embedded in the thylakoid membrane and a transmembrane channel that carries out facilitated diffusion of protons across the membrane. So this is a channel that will bring about facilitated transport of protons across the membrane. The other portion is called CF1 and protrudes onto the outer surface of the thylakoid membrane on the side that faces the stroma. Same thing, like the plug that we have seen in the mitochondrial membrane. This also is jutting on from the thylakoid membrane. The breakdown of the gradient provides enough energy to cause a conformational change in the protein particle of the ATP synthase, which makes the enzyme synthesize several molecules of energy-packed ATP. Chemiosmosis requires a membrane, a proton pump, a proton gradient, and ATP synthase. So chemiosmosis theory requires a membrane, proton pump to create the gradient, electron carrier, and a proton gradient, and ATP synthase. 4 things. Done, no? I think we have discussed most of the things. Where are the ATP and NADPH used? All of that story we know. Energy is used to pump protons across a membrane to create a gradient or a high concentration of protons within the thylakoid lumen. ATP synthase has a channel that allows diffusion of protons back across the membrane. This releases enough energy to activate ATP synthase enzyme that catalyzes the formation of ADP to ATP. Along with the NADPH produced by the movement of electrons, the ATP will be used immediately in the biosynthetic reaction taking place in the stroma. The dark reaction or the light independent reaction, Calvin cycle, that is happening in the stroma, responsible for fixing carbon dioxide and synthesis of sugars. All right. Clear? Light reaction. Now you can relate both the respiratory pathway in mitochondria and the light reaction here. Operates on the same principles. Okay? Okay. So we will stop discussions now. Any questions related to this you ask. Otherwise another 15 minutes you can continue your own studies.